In this lecture, we're going to carry on talking about cellular respiration, and in particular, we're going to talk about the second phase of cellular respiration, and that is pyruvate processing, or PP for short. So, pyruvate processing happens inside an enzyme complex known as pyruvate dehydrogenase, and this enzyme complex is found within the mitochondrial matrix. So what happens after glycolysis is that the two molecules of pyruvate that are produced for each molecule of glucose move across the outer membrane of the mitochondrion and then through small pores and enter the matrix using, so remember the matrix, enter the matrix using a membrane protein known as a pyruvate carrier. And this is within the inner membrane of the mitochondrion. In order to get pyruvate into the mitochondrial matrix, ATP is required. So it's an active process. Pyruvate is moved across. So remember, it's the whole adage of spending money to make money, or in this case, spending energy to make energy. So the movement of the pyruvate requires ATP in order to get into the mitochondrial matrix. Now, there are a number of stages within pyruvate processing, and the whole process is laid out here, as you can see in this diagram. Like I said to you in the previous lecture, I'm not too worried about exactly how this is written, but I do want you to understand each of the processes that we're going to list down here, and each of the stages of pyruvate processing are important. So remember where it happens within the mitochondrial matrix. And what we see as the first stage is that the pyruvate from glycolysis reacts with a compound called coenzyme A, CoA, to produce what is known as acetyl coenzyme A. And basically what happens here is that there's a transfer of an acetyl group to the substrate. What is the substrate, you ask? Well, that is the pyruvate in this instance. So that's the first step in pyruvate processing. The second step is that as this pyruvate is being processed, which is in this section of the diagram above over here, one of the carbon atoms in the pyruvate is oxidized. Remember what happens when there's oxidization? There's a loss of electrons, and an NAD plus is reduced to NADH. In the third step, acetyl coenzyme A is the final product, all right? And this reacts with oxaloacetate, as you can see over there, to form citrate. And this citrate is the very first component of the Krebs cycle, which we will talk about in the next lecture. Right, so just like glycolysis, pyruvate processing is regulated. Remember we said that almost all processes, in fact most processes within a cell are regulated. And just like glycolysis, the pyruvate processing tends to stop within a cell when ATP is abundant. Again, that makes sense because when ATP is abundant, there is no need really for cellular respiration to proceed. So pyruvate dehydrogenase changes its shape when there's a lot of ATP in the cell. So in essence, the ATP binds to the pyruvate and changes its shape. This means that it becomes phosphorylated when it changes its shape, and the entire process of pyruvate processing stops. Importantly, the rate of phosphorylation increases when acetyl coenzyme A and NADH are in high concentration. So that's effectively the opposite. So when we don't have a lot of ATP in the cell, we see that there's increased phosphorylation to try and get the cell to produce more ATP, ultimately through the process of cellular respiration. So, if we return to our summary table, our overview of cellular respiration, we've now completed 2 of the 4 stages of cellular respiration in lectures. The second one has been the stage of pyruvate processing, and you'll see that the two pyruvates that were derived from glycolysis are what goes into this process, along with two NAD pluses, as well as two coenzyme A's. What are the products? Well, we have two carbon dioxide molecules. The NAD plus is reduced to NADH, and we have two acetyl coenzyme A's. Unlike glycolysis, this process, pyruvate processing, takes place within the mitochondrial matrix. And in the next lecture, we'll talk about the all-important citric acid or Krebs cycle and electron transport to produce the final and most important products of cellular respiration.